Welcome back, everyone. Thank you for watching us through Zoom and YouTube. And here we are on day 4. Over the past days, we've explored concepts, case studies, and even ethical dilemmas. Now it's time to connect the pieces: computer vision, segmentation, molecular networks, and see how they work together to create robust, interpretable, and impactful medical solutions. Think of today as the bridge between the science and the systems. Today's set of lectures begins with Professor Dr. Tim Kacprowski returning once again to share his expertise at the intersection of biology and computation. This time, he'll show us how molecular interaction networks can make artificial intelligence both more robust and interpretable. A perfect example of how systems, biology, and AI can work hand in hand. Please join me in welcoming Professor Tim Kacprowski.
Thank you. Good morning, everyone. Today we're going to talk about how molecular interaction networks will help to make machine learning robust and interpretable. And that's actually a neat tool to use if you kind of want to circumvent all the math stuff we talked about last time, right? So to that end, I'm first going to tell you what's wrong with gene signatures. So I'm going to if you want, toot a bit the same horn as Antonio did when he told us that different approaches for identifying differentially expressed genes are not very consistent, and there might be something that we're doing wrong there. I also think there's something that we're doing wrong there, but we approach this from a bit of a different perspective. And then, of course, I want to tell you how you can use networks to do better than without networks. And first, just a very brief introduction into molecular networks so that you know what we're talking about. And then I will show you an approach that other people developed for a supervised case, and an approach that we developed ourselves for the unsupervised case. And as a last point, I'm going to show briefly a tool that we recently developed to actually construct networks. Because one question is, of course, where do we have all these networks from, right? Okay, so what's wrong with gene signatures? Well, gene signatures are basically just a collection of genes that you use as biomarkers, for instance, in cancer diagnosis or for other diseases. Then you look at kind of the expression of those genes, and you can say, okay, if this and that gene is very highly expressed, then the patient likely has this or that disease, or you need to treat the patient in this or that way. And this goes back to the time when I spent a postdoc in Jan Baumbach's group at the time in Denmark. We had Simon in this group as a PhD student, and we regularly had these it was a winter workshop, not a summer school, because it was in Scandinavia, I guess. But Simon then talked about novel methods for discovering gene expression signatures for breast cancer outcome. Now, how do we find those gene signatures very generally? The idea is that you have a group of patients, and then you want to divide those groups in the simplest case into 2 subgroups, where one group of patients has a good prognosis and the other group has a bad prognosis. And we do this usually by statistics, so you split the patients based on your favorite multivariate statistics, let's say PCA, which gives you 2 groups. And then you compute a Kaplan-Meier statistics between those groups, or a log-rank test or something, if you want to have a p-value. And then you see you have a blue group and a red group. And the blue group, are you familiar with Kaplan-Meier plots? So basically, you start with all your patients, and then time flows to the right here on the x-axis, and whenever this curve goes down, a patient in that group died. Okay, so that means if you compare these 2 groups, the red group dies off very faster than the blue group. So if you're a patient, you probably want to be in the blue group, right? Why are we interested in this? Well, this helps, for instance, to prioritize treatment of those patients, right? So you could argue that those are high-risk patients that need more intense treatment, for instance, than patients in the blue group. The data, if we have transcriptomics data, you can usually represent in these kinds of feed maps, which is just a colorful table if you want, right? You have, for each row is a patient, and each column is any given gene, and the color shows you the expression of that gene. And since this is very easy to do, there are many, many signatures for breast cancer already out there in the literature. And just to give you a few examples, and if you're watching the recording, you might want to skip ahead a bit. If you're online, now would be a good time to go and maybe grab an ice cream or a cup of coffee, because there are a lot of gene signatures, and we are only talking breast cancer, right? Are you still good on drinks, or do you want to go grab something? We have time, right? Very good. Because there are a lot of, and we're only talking breast cancer, right? And there's many other cancer types, and there's many other diseases. So there's really a ton of signatures out there. So Simon here thought, well, why don't we add 3 additional gene signatures to the pool? Because those things, seem to be useful, they're nice to publish, and people want them, so let's come up with 3 new signatures. Of course, if you want to do this, you need some data, and as a data scientist or computational biologist, where do you usually get your data? Well, you go to the internet and download some publicly accessible data. For cancer, there's the Cancer Genome Atlas, where you can download really a lot of data via the CBio portal here. And there, Simon found RNA-seq and clinical data for 526 patients, including their survival times. So he had enough material to actually identify those signatures, right? Because in most, let's say, real projects or new projects with fresh data, it's actually hard to come up with that many patients. So this is a really good data set for this. Now, those gene signatures also always have very funny names. And the first signature Simon called Arnold 255. Which kind of statistical approach did he use? He did something he called time series mortality statistics from an Austrian cohort. Sounds fancy enough, right? What did he actually do? Simon went to the website moviebodycounts.com, where you can query for movies or actors or something, and then you get, as a result, a body count of that movie. That is how many people die in that movie. Like not real actors dying, but people dying in the movie. So you see here the numbers of kills in those Arnold Schwarzenegger movies. And just as an aside, has anybody any idea what's still up to date, at least a few weeks ago when I last checked, the movie with the highest body count ever? People usually guess something like Terminator, whatever version or so, right? But it's actually Lord of the Rings, The Return of the King. That's some food for thought. Anyways, so now we have those numbers here, right? What do we do with those numbers? Well, we add 255 because we can, which gives us these numbers here. Now, how are we any closer to a gene signature with this? Well, if you remember that we have our data in such a table or matrix, you can now just use those numbers here as indices for the genes you put in your signature. Okay, so what you do is if you have 343 here, you take column number 343, let's say that's this one, and then you use the gene that happens to end up in that column as one gene in your signature. And then you use those genes and you do all your statistics, right? You do your PCA or your regression or whatever you do. And you define two groups of patients based on the expression of those genes. Does that make sense? No, right? But it works. You get two groups of patients with a significant difference in survival by selecting genes based on body counts in Arnold Schwarzenegger movies. Like, what the heck is going on here? That's exactly what Simon thought, and we have two more tries, right? So let's come up with a second gene signature. What do people, especially in the biomedical field, like to do if they want to find out if stuff really is like they think it is? They look into the literature, right? And that's what Simon did. Simon, for his second signature, went into literature mining. Of course, if you want to do literature mining, you kind of have to restrict the space that you're looking at because there's too many papers out there for anybody to read. So he just looked at the years 1997 to 2007. And the second signature he called HP40. So how did literature mining work for Simon? He Googled how many words are in which Harry Potter book, which gives you these numbers here. And then we multiply these numbers by 40, modulo the number of genes, again, because we can. And we do the same thing as before, and guess what happens? You do find two patient groups with the significant difference in survival. This is at least scary, right? So let's see if we really want to be sure, and if we, test whatever we see here and want to make sure that we're not just doing something stupid. We do stuff in animal models, right? We do experiments and we check in an animal model if really they show us happening what we assume to be happening or what we predict in our experiments or whatsoever. So Simon went for an in silico mouse model for his third signature, which he called Pikachu star 5. How did the in silico mouse model work for Simon? He used the characters that make up the name Pikachu, converted them to the ASCII code, which is this here, multiplied them by 5, and again extracted those genes. And guess what happens? You do find two patient groups with a significant difference in survival. So what on earth is going on here? Why does this work? I mean, neither Arnold Schwarzenegger, nor Harry Potter, nor Pikachu should have anything to do with breast cancer. Does anybody disagree? Okay. So what's going on? Why does this work? Well, there's two things that kind of make this work on a very abstract level. One is 500 patients are a lot, but do you know how many transcripts we look at in these kinds of studies? It's like roughly 25,000 or something, right? So you have 25,000 features to find two groups among 500 samples. There's many, many ways to find two groups if you have that much fewer samples than you have features, okay? So it's a very ill-posed problem in that sense. The second point is there is obviously no biology in there whatsoever, right? We only do statistics. And if you do this in a less funny but more systematic way than Simon did, and you look at, for instance, MSigDB, which is a database that contains all kinds of signatures for all kinds of diseases, not only breast cancer, you see, for instance, this plot here where the blue curve gives you real signatures that are in this database, and the red curve gives you random signatures. Could be formed in a way that Simon did, could be by just randomly selecting genes without having that much fun. The x-axis gives you the p-value, actually the negative logarithm, so the further left you are, the more significant that signature is. And your usual threshold of significance is this gray line here. Now what you see is that you find more real signatures than random signatures that are not significant, and you find more random signatures than real signatures that are significant. And this should not be that way if what we do makes sense, right? Because if what we do to find those signatures makes sense, then those signatures should be significant. But they're not. We can just by randomly picking your favorite genes, or, I mean, if it's your favorite genes, it's not random, right? But Just as an aside for randomness, have you ever tried to randomly put stuff on a pizza? Because if people try that, they usually end up with stuff uniformly distributed on the pizza, but not randomly. It's an important but little distinction. Anyways, so there's something wrong with those signatures, right? However, they're still used in the clinics. They still kind of make sense statistically, right? They work to diagnose patients, to predict the outcome, but only statistically, not biologically. They don't tell us anything about what's going on the molecular level. They just allow us to do some statistics that change how we treat patients. So you could say, so what? It works statistically. We could be happy and still, keep doing this. And statistically speaking, patients would still be better off than if we would not be doing this. But are you familiar with this concept, the number needed to treat? It is the number of people you need to give a certain drug to in order for one person, or to be certain that one person you gave that drug to actually has a benefit from that drug. And those numbers are quite high here, right? So the blue people are those for which we are sure, like not in the sense that you know who this is, but statistically speaking, for this person, this drug will work. For the red people, the drug will not work. And why is that? Because for many drugs, we don't know why they work or how they work. And for many diseases, we don't know what's going on a molecular level. And how we find drugs that work is, again, most of the time statistics, right? We use these randomized controlled clinical trials and whatnot, which is basically the same on some level, at least what Simon did for the gene signatures, right? You divide your patients into two groups, and then one group gets the drug, the other group doesn't get the drug, and at the end you check if on average the group who got the drug does better than the other group. So pure statistics. And arguably this should change, right? We want drugs to work. Because you could say, well, that's nice for the pharma industry, right? You can sell many drugs and still do some good for some people, but you have to keep in mind that those drugs also have side effects. So it could actually be that you leave the world a worse place by giving that drug because there are many people who potentially suffer from the side effects without having any benefit. So how can we get out of this? And as you probably guessed, we can do this, for instance, using molecular networks. So this is not networks for deep learning or anything, right? This is really kind of a little bit of graph theory, modeling how molecules interact in biology. I mean, networks are everywhere. We have social networks, economic networks. Biomedical networks also are the one that are most interesting for us now. But there's many things out there in the world that you can frame as a network or a network-related problem. Why do we study those in biology? Well, because, interestingly, in biology, seldomly 2 + 2 = 4, but rather 3.8 or 7, I don't know. So you cannot really understand what's going on in biology if you just look at individual molecules and what they do. You have to look at kind of the whole system that you're studying, right? Be it a human, be it your favorite yeast cell or whatever. So you need a bit of a more holistic perspective. If we talk about molecular networks, we are basically modeling the interactions between, for instance, genes or transcripts or proteins or metabolites in networks. And if you have a different favorite biomolecule, then you can also use that in a network, right? So those things could look something like this, where the nodes here are proteins or metabolites, and then the edges between the nodes tell us what kind of relationship connects these two. It could be some metabolic reaction, could be some gene regulation, could be some cell signaling, or some protein interactions, right? The important thing is this gives us some biological meaning, right? Because the interaction here between B and D means that those two proteins actually interact in some biological process to make, so to say, some biology happen, right? Nothing will happen if you just have a single protein sitting around somewhere, but this protein needs to interact with other proteins or with other biomolecules to actually observe some biology. And that's what we can nicely model with these networks. Just for a few examples, you can see here something like the yeast proteome, so all proteins in yeast and how they interact with each other. This is an excerpt of the human proteome. So for now, you just see some hairballs and they won't tell you much, right? If you've ever been to the office of any person working in metabolomics, you've probably seen this hanging on the wall. This is a metabolic network. This is kind of a zoom in for a metabolic network, which we can model in different ways as a graph. Just so you see that this is really a very flexible tool that's not restricted to a certain level of biology. Here we have a gene regulatory network with different kinds of edges, so we see which gene increases the expression of another gene or decreases the expression of another gene. Here you have a neural network, but not an artificial neural network, but a network of the neurons of C. elegans. So this is a brain, if you want. You can also go beyond biomolecules, and you can make networks of human diseases. So here every node is a disease, and the edge tells us that there's some overlap in terms of the involved genes, for instance, right? So you find genes that are involved both in, what's that, deafness and this no-name disease here. This edge means that they're the same genes involved in both diseases. So this helps you a lot if you are interested in things like comorbidities and so on. You can make networks of drugs, and you can make networks basically of anything, right? So these are just so-called metagraphs, which kind of define what kind of nodes you can have in a network and what the interactions between those nodes are. So you see this is really a very flexible approach. You only use or need nodes and edges, but you can basically model whatever you want. And of course, people use this to understand and investigate diseases, right? Especially in the multi-omics context, where you have networks on each omics level, and then you can connect those networks based on what you know between omics processes, and then you can use those to actually, diagnose diseases, find biomarkers, do some graph-related deep learning, or whatever you want to do with these networks. So how can we use this in a supervised setting? You've already heard about random forests before, right? But just as a brief recap, a random forest is basically a collection of decision trees where each of those trees sees a different subset of your samples and a different subset of your features. So there's two things that are randomized in those forests. One is the samples and the other is the features for each tree. This, you can plot those trees also as kind of these decision surfaces here, which means that each tree gives you a different decision surface. If you overlap those, then you basically have a random forest. That's the main concept, okay? Now, how can we use this, or how can we get molecular networks in random forests? It's a very easy, but as you will see, also very powerful idea that Simon and Jan came up with back at that time. The idea is, instead of selecting the features for each tree totally randomly, we select the features in such a way that they form a subnetwork. So if we want to use a random forest for some human disease, let's say we use the human interactome, so protein interactions in humans. And then the first node in this tree will be some random protein, let's call it V1. The next node this tree can choose for partitioning the samples has to be a neighbor of V1 in our protein interaction network. So we are only allowed to choose V3, V5, or V2. The other nodes are not available for this tree at this time. And if you grow the trees in that way, in terms of the features you select, then you make sure that each tree in your random forest is, by construction, also a subnetwork in your protein interaction network. Does everybody see that? Okay. It's a very simple idea, right? But it actually works very, very well, as we see here, for instance. So there's different approaches. GF stands for Grand Forest, which is this graph-restricted random forest that I just explained. And they tried this on 5 diseases: breast cancer, lung cancer, ulcerative colitis, Huntington's disease, and amyotrophic lateral sclerosis. And you see the enrichment of disease-associated pathways in the important features you get out at the end. so in your signatures, if you want. And the higher up this box is here, the more enriched in disease-associated pathways your features are. That means, according to everything we already know so far, those features are more relevant, biologically speaking, okay? And this black line here shows the threshold for significance. So everything that's below this line does not give you any significant relationship to the disease in terms of the features you get in the signature. So all this Arnold Schwarzenegger, Pikachu, and Harry Potter stuff would be also below the black line. Now, Grand Forest is the only approach that gets above the black line, right? So it's the only approach of those evaluated here that actually makes sense in terms of biology. It's a little bit different for Huntington's disease. There also Grand Forest can help us much. Any idea why that's the case? Maybe the medical people know. So cancers are arguably very complex diseases, right? Huntington gene, if that gene is mutated, you will have the disease. If that gene is not mutated, you will not have the disease. So why would we need networks to find something out about that disease when there's only one gene that we know is causal for the disease? So you can see this here as kind of a negative control, right? I think back to permutation tests, we would expect that this does not get significant. Everything else would be strange. Or we would have found something about Huntington's disease that nobody knew before, but that's rather unlikely, right? So this is a negative control, so everything kind of makes sense. But you see that Grand Forest is the only approach that actually gives you biologically meaningful and interpretable signatures. It's only also the approach with the most robust signatures across those diseases. So here, the higher up the box is, the more robust the signature is. That means if you perturb your data a little bit and rerun the whole thing, you get the same set of features. So for most approaches, you get very different sets of features when you rerun this on slightly perturbed data. That is adding some noise or something. And for Grand Forest, these signatures are actually more stable. Again, Huntington's is kind of the exception here, which makes sense for the same reason as before. Now, people usually want significantly differentially expressed genes, right? That's why we have all these approaches that also Antonio mentioned earlier. But if you look at the reference pathways for those diseases, so those pathways that we know according to literature are involved in the diseases, and you check how many of the genes in those pathways, the red ones here, are actually differentially, significantly differentially expressed in many studies out there, it's only a fraction, right? So many of the genes that are involved in these pathways are not significantly differentially expressed, which means significantly differentially expressed genes might be a nice starting point if you investigate some new phenotype or some new disease, but it should not be your ultimate goal because it only gives you like a fraction of what's actually happening and what's actually involved in a disease, speaking from a molecular biology perspective. So this was the idea for the supervised case, and then later we also came up with the one for the unsupervised case, meaning clustering, right? So again, we have a data set here, genes and patients. We use networks, can be protein interaction networks, gene regulatory networks, or your other favorite network. And then we want to do some bi-clustering here. So we cluster genes and patients at the same time. And we want subnetworks that explain this clustering. So basically subnetworks of genes that show different behavior in these two groups of patients. We developed this approach called BiCon, bi-clustering constrained by networks. And the first thing that we realized is that this is very robust to noise, right? So we can have up to 40% of noise in the data, and it still works almost perfectly, right? And this also goes back to, remember when I told you in the first of my lectures, or the second lecture of the school, that noise is usually not a problem for us, right? It's even less a problem if we use networks. And why? Well, because at least if everything we assume is correct in an ideal world, then these networks encode biology and not technical measurements, right? So you will have noise in your technical measurements, but the biology you put in through those networks will be stronger than your technical noise, which is why this is not a problem here. But let's see if it can also handle batch effects. So that's something that we very frequently encounter in biomedical data, right? If you measure something here and you measure something in Germany, for instance, you will get two very different results simply because you have different machines or different people operating the machines and so on and so forth. So we simulated some data with a really strong batch effect here. You have the orange and the brownish batch. And the goal would be to distinguish the gray from the purple patients, right? So to kind of put all the purple rows together and all the gray rows together, but not the orange and brown rows. What people usually do is that they kind of normalize this or try batch effect correction methods. If you do this, the whole thing looks like this. So we nicely mixed the two batches here, but we're nowhere closer to putting all the purple and all the gray together, right? So this, in this case, doesn't work because the batch effect is simply too strong. But if we use SpyCon and we let some ants run around on our protein interaction network, I will show you that we can actually solve this. Now, why ants? Well, we use a technique here called ant colony optimization, which is kind of a bio-inspired computing scheme. In brief, what we do is we construct a joint network based on the expression and the protein interactions. And then we use this ant colony optimization in a local search procedure to find some potential solutions and then check how well these solutions work. So this is one of those problems where it's generally very hard to find a solution computationally, but once you have the solution, it's easy to check if it's a good solution or a bad solution, which means you can just, well, not that randomly, but you can kind of just explore the solution space and suggest different solutions and then check if they work or not. This is easier than trying to find the best solution from the start, okay? Did you ever have ants in your living rooms or bedrooms or something? And have you observed them for a while? Then you know that they kind of, with time, form these ant streets, right? Does anybody know how this works? Like why they form these streets? Well, if they leave their nest, they kind of wander out randomly to find food. And when the ant finds some tasty food on the way back, it drops pheromones. And then the next ant that wanders out, I don't know if they technically smell, but let's say they smell. They smell these pheromones and then they follow the same route. And once enough ants have dropped enough pheromones, everybody will follow that one route, right? That's what's happening in your living rooms. Technically, the same happens in a computer, except that now the ants don't drop pheromone and smell it, they just do some math, which we don't have to worry about. The process is the same, right? If we let them roam around in the networks for long enough, they figure out which are good ways in the network. And then the way we do this is that each patient, those are the P's here, will be treated as an ant colony, and we let ants start walk around in the network from that patient, and those are the genes or proteins. And then, for instance, ants from this patient will predominantly walk around on genes G1 to G5, because they like those genes for whatever reason. You do the same for patient 2, so a different ant colony, ants walking around, mostly on G1 to G4, and so on and so forth. P3, again, predominantly walking on those genes, P4 and P5. And then we can check which genes did the ants walk at, or which genes did the ants like, and we can use this to cluster the patients, right? So all the ant colonies from patient 1, 2, and 3 liked very much those genes G1 to G4 or G5 here. So this could be a good cluster of genes, and this could be a good group of patients for those genes, right? So that's what we do. And then we just drop gene 5 because it's kind of in the intersection here. So we actually get a small network here of those genes who are important for those patients, and a small network of those genes who are important for those patients. And then we have clusters of genes, so 2 groups of genes and 2 groups of patients. This is how the ants, or how we find out if it's a good solution or not, right? Basically, it just means we want some connectivity here so that this should be connected, but it should not be too large, because the trivial solution would be to use all genes, right? We don't want that. And this just tells you that the expression should be different between those genes in this patient and in this patient. So it's kind of going back to differential expression, but we just say we want the difference of expression of those genes between those patient groups to be high. And if we do this, we can almost perfectly reconstruct gray and purple groups of patients here. There's one that we miss, but well, we're not living in an ideal world, so perfect would be too nice. It's almost perfect, right? Green and blue now are the clusters that we get out of this approach, and purple and gray, again, are the two groups of patients that we wanted to be clustered. We also get those networks here, which tell us about the genes that are responsible for this clustering of patients. Now, this was simulated data. It also works with real data. So here, we're distinguishing luminal from basal breast cancer. Goal would be to group all the purple and all the yellow patients. Almost work perfectly, right? There's some that are off, but again, almost perfectly. Now, if you ever worked with breast cancer expression data and tried to distinguish luminal from basal breast cancer yourself, you would probably now say, come on, that's an easy problem, right? Anybody can do this, you don't need networks. And you're right, it's easy. But there's a harder problem, which is distinguishing luminal A from luminal B breast cancer. Lo and behold, we cannot do that at all. Even with this approach, it doesn't work, right? Like yellow and purple are all over the place. So why am I telling you this? Well, the nice thing here is, because of the networks, we have some biology in there, right? And those two groups that we get, albeit they're not luminal A versus luminal B, they are groups with an important biological difference, meaning, in this case, the microenvironment score, so the immune infiltration of the tumors, which, oncologically speaking, is something that you really want to know if you want to treat your patients, right? So we do not find what we asked for, but we find something that's very meaningful and very biologically relevant without even asking for it. Isn't that cool? Okay. So take home here, you can find biological differences that are relevant, and you don't even need to know what you're looking for, right? Because we put in this biology based on these networks. So just briefly, as a third route approach that we developed to actually construct these networks, or some kind of networks, it's called BoostDiff for regulatory network inference, and specifically we want to infer the difference in networks and gene regulation between two conditions. So we're not only interested in which genes are differentially expressed, but we're interested in how does the regulation of gene expression change in two conditions, okay? There's an approach out there called GeneE3 for inference of regulatory networks, which I would say is still the champion approach. So if you want to infer networks, you probably use GeneE3 because it works surprisingly well. The idea behind it is based on regression forests, which are very similar to random forests, just except that you classify something here, you do a regression. So you want to predict numerical values. And the idea is that for all the genes you have, you do this kind of cross-validation scheme here where you try to predict the value of one gene based on all the other genes. And you do this for all the genes. And then at the end, you get these feature importance values that tell you, okay, for instance, these two genes here were important to predict the expression of this gene. And if they're important to predict the expression of that gene, then you could argue they might be regulating that gene, right? Which is why you would infer a regulatory connection between those important genes and the gene that was the target for your regression. And by this, you can look at all the importances here and then kind of order them and construct a network of this gene regulation. And this works really very nicely. And those tree-based approaches are usually nice because you can have very simple ideas to change those which have a very high and practical impact, right? As we saw with Grand Forest, a very simple idea to just change what features the trees can choose when we train them with a huge impact. And here we had a similarly simple idea in a different direction, because the way you usually train those trees or you learn those trees is that you have your samples of the two conditions, let's say blue and red, and you have some feature and some threshold so that you decide whether the sample goes right or left through this tree, right? And then you try to end up with leaf nodes that are consisting only of blue or only of red samples if you do a classification. The way you can measure this, like if the split actually improves your situation or not, is by this impurity here. So you take the difference of the impurity you have before the split and the impurities you have after the split. If you can reduce this impurity, then this would be a nice split, right? So if here you have a mix of the two conditions, and here you have mostly red, and here you have mostly blue, so this would be a nice split of those samples. Now, in regression, this impurity is measured kind of as variance, right? How much variance you have in those values that you're trying to regress on. Since we have those two conditions, what we can do is that we compute the improvement of the split separately for these two conditions, this variance in the case of regression, right? So we kind of let all the samples percolate through the tree, and then we do not only check the variance over all samples, but we separately check the variance in the samples of the blue group and the variance in the samples of the red group. This allows us to find or to do splits that work very well, for instance, for the blue group, but don't work for the red group. Or the other way around, do splits that work very well for the red group, but not for the blue group. Why would we want to do something like this? Well, if we want to find different regulatory mechanisms in these two groups, we want to find regulation that is only taking place in one of the groups, not in both groups. Right? So we're actually looking for features that allow us to do a nice prediction in terms of the regression, for instance, in the blue group, but that do not work well in the red group, because then it would be clearly something different in the two groups. Do you see that? Okay. So this is kind of the adapted workflow of BoostIF. Looks very similar to Genie 3 because it essentially is, except for this new way of computing how well your split works, so the new way of learning your trees, right? And then we get this differential network here at the end, where we have some regulations that are stronger in condition A and some that are stronger in condition B. And again, just to show you that this actually works, if you're interested in COVID-19 or Crohn's disease, then you would now be very amazed if you see these networks, because those actually biologically make sense. So we do find differential regulatory processes here that are different in COVID-19 patients versus healthy controls, right? Or in Crohn's disease patients versus healthy controls. If you're not so much into COVID or Crohn's, then these are two hairball networks, and well, okay. But to show you that it works principally, what you see here is the correlation between pairs of genes. And the pairs of genes are selected in different ways. So here we have results from BoostIF when the disease is the target condition or when the control is the target condition. And here we have differentially expressed genes. And here we have randomly selected genes or edges in the networks. You see, in the random case, most of the correlations are around 0, and you kind of have this almost normal distribution around 0 here, which means that randomly selecting genes doesn't give you much information about the correlation, so about the difference in regulation in these two conditions. Okay. If you look at the differentially expressed genes, it gets a bit better because we have this almost bimodal distribution here, so you do find some correlations, which means you find some actual difference in the regulation between these two conditions, but not much. It looks very similar to just randomly selecting genes, right? If you do BoostIF, you see that you have a very clear bimodal distribution for your respective target conditions. So now you actually find edges that should be present and that biologically happen only in one of the two conditions. So you actually find what you're looking for, the real difference in regulations between these two conditions. Right? Because the correlation is only happening in one condition, but not in the other condition. So this regulation is relevant in the disease, but it's not relevant in the control group, or the other way around. This works for COVID and for Crohn's. In case you're more into little things, it also works for S. aureus in different stress conditions. But if you're not into SREs, then just think of this. That shows you that it works principally, right? All right, so many people have actually done the work, right? I already mentioned Simon and Jan, who did the Grand Forest stuff. Olga Lazareva was the kind of main developer of Bicon, and Markus List and Johanna Galindes were involved also in the BoostDiff approach. And I have not prepared any IPython notebook, whatever, for you to solve, but if you're interested and if you want to check this out, you can play around with these 3 tools. There's also some example data you can use on the websites or in the GitHub repository, and then see if the results make sense for you and if you understand what's actually going on there. And if you have any questions about those, of course, feel free to reach out anytime. This has been my group at some point in time, some collaborators, and as a conclusion, I would argue that artificial intelligence is only intelligent when used intelligently, right? So don't go out there and ask ChatGPT, hey, what's the difference in gene expression between my 2 groups or something. As an aside, again, there has been recently a paper that shows that those language models can very well predict the position of planets in our solar system at any given time. But if you ask them why the planets are there, they come up with all kinds of bullshit. So that's nowhere near explainable or interpretable. Molecular networks are a very versatile way to push some biology into your AI or into your machine learning, as I hope I've demonstrated, at least for some examples. They do come with their own problems though, but I guess this is nice because this still keeps us busy. So thank you very much.
Thank you so much, Dr. Tim. We have a few questions from the Zoom meeting. Would you like to answer them? Can these interpretable models be used in real-time clinical decision support, or are they still more research-focused, especially in your university?
They're definitely more research-focused for the very simple reason that I think it's tough to get a real-time whole proteome or whole transcriptome measurement, right? I mean, those things take time just to measure from the patient. So I think without significant improvement in terms of measuring technology, we are rather in a research-oriented setting here than in a real-time clinical setting.
And how do you ensure computational efficiency when scaling from small curated networks to large heterogeneous data sets?
Well, I want to refer to network-based approaches come with their own problems though, right? So very frequently those things don't scale that well. So it's tough to do that on at home on your laptop. You really should have some kind of probably small cluster in your basement if you want to go through with that. But if you have any ideas for how to make it more efficient, let me know. Happy to collaborate.
Thank you so much for your answers. If there are no comments from the public on site, then I guess we're done. Please give an applause again for Dr. Tim. Thank you so much for your presentation.